our Keynote environment, and you will already see Dr. or Professor John Mathers in the picture. He is a professor of human nutrition and the director of the Human Nutrition Research Center and director of the Center for Healthier Lives at Newcastle University in the UK. His major research interests are in understanding how eating patterns influence risks of age-related diseases, including heart disease, diabetes, dementia, and bowel cancer. And he uses genomic and epigenomic approaches to understand the mechanisms through which nutrition influences cell function and ultimately health, which of course is very much aligns with a lot of the research we have funded as well from the JPI HDHL. And today you will, guide us through the advances in nutrition and health research over the past 10 years, but you will also provide a glimpse into the future, which I think will also be very interesting for the audience. So I will just keep my mouth and give the floor to you, and I will be back. I will put off the microphone and the camera, but I will be back for the discussion. So,
perfect. Thank you very much indeed. So good afternoon, everyone. I'm delighted to be at this great JPI 10th anniversary conference. So what I'm going to give you is very much a personal view of some of the advances in nutrition and health research over the last decade, and some hints as to where things might move in the next 10 years. I'm going to start with some obesity-related issues, and that's clearly been an area where there's been major advances and advances in understanding diseases such as diabetes and dementia. Then I'll move on to talk about nutrition and the gut microbiome, which has been one of the most exciting areas of research and one where we're still making very rapid progress. I'll also speak a little bit about personalized nutrition, which was on the agenda 10 years ago, but there had not been very many studies, lots of review articles, but in the last 10 years, we've had a number of intervention studies to test the idea, and I'll discuss some of the outcomes of that work. And of course, all science depends on having good tools and technologies, and I'll talk a little bit about how we've developed better tools and technologies for nutrition research, and then finally look to the future. So starting with obesity, and as I guess all of you know, for the last 30 or 40 years, the rates of obesity have been climbing in most parts of the world. And along with that increase in obesity has been an increase in a range of non-communicable diseases. And I guess in the last 10 years, we've really begun to understand how excess fat drives these common diseases. So diseases that might in the past have looked to be quite separate, we now realize have common elements in their etiology. So over the last 10 years, for example, we've begun to understand much more clearly how obesity drives the risk of a whole range of cancers, and there are now at least 12 or 13 cancers which are strongly related to how obese we are. I'm going to pick just two examples from this, and I'm going to start with type 2 diabetes. And this is one of the conditions which is most closely linked with adiposity. And as we've got fatter across the world, so rates of diabetes have increased. And now in the UK, nearly 5 million people have type 2 diabetes, and there are well over 400 million people globally with this condition. And I guess if you go back 10 years, most people would have thought that a diagnosis of type 2 diabetes was a life sentence. In other words, you would have to live with that and live with all the consequences of type 2 diabetes. But there have been dramatic turnaround in our understanding of diabetes, and now we know that it's not necessarily a life sentence. I'm going to start by showing you some work done locally by Roy Taylor and the group in Newcastle. And what this provided was proof of principle that rapid weight loss can reverse type 2 diabetes. So what you see on the slide is changes in blood glucose concentration over an 8-week study. A raised blood glucose concentration is a key marker of type 2 diabetes. And what you can see is that within 1 week of being put on a very low energy diet, the participants' blood glucose concentration had got back down to normal and has stayed normal throughout the rest of the study. Now, the important thing is that that change happened really quickly, long before the people had lost appreciable amounts of weight. So something was happening that was related to weight loss, but wouldn't be measured by just getting on the bathroom scales. This then led us to the DIRECT study, the Diabetes Remission Clinical Trials. This is a much bigger study carried out in primary care to test the idea that effective weight management could produce sustained remission of type 2 diabetes. We carried out this study in Scotland and in the northeast of England, and the intention was to enable people to lose large amounts of body weight and to sustain that weight loss. And our target was to lose 15 kilograms of weight loss. Importantly, the people who went on the study were taken off their diabetes medication. So what we were looking for was a nutritional intervention to cause diabetes remission without having to involve the use of pharmaceutical agents. What I'm showing you here are the data after 1 year of the trial. The vertical axis is the proportion of the participants who were now free of their diabetes. So at the end of the first year, those people who had managed to lose at least 15 kilograms of body weight, 86% of them were now free of their diabetes, and they were off medication and felt so much better. And there's a clear dose-dependent improvement in diabetes remission the more weight the people were able to lose. So what's going on here? Why does weight loss have such dramatic effects? Well, we think that part of the explanation is what happens deep in the body. So what we're seeing here are MRI scans through the abdomen of an individual. And on the left, you see the individual when that person had type 2 diabetes, and their liver shows up bright green because it's full of fat. 36% by weight of the liver was fat. On the right-hand side, you'll see the individual after weight loss, and now the liver's looking dark blue, and that's because almost all the fat has been removed from the liver. They're now down to 2% liver fat. The area in red around the outside is subcutaneous body fat. And although they have lost some subcutaneous body fat, very little by comparison with the amount of fat that's been removed from the liver. So we think that the key to diabetes remission is this removal of fat from the liver and also from the pancreas. And that removal allows normalization of the body processes to control glycemia. So when we remove that fat from the liver, and particularly from the pancreas, we normalize glycemia and people are free of diabetes. We followed up the individuals in the DIRECT trial for 2 years, and we've sustained that remission of their disease. So the second example of an obesity-related disease is dementia. And this is another disease which is climbing rapidly. And it's predicted that by 2050, there'll be a 3-fold increase in the number of people living with dementia around the globe. Those increases are happening all over the world. They're happening in Europe and elsewhere. But the biggest increases are in the less developed parts of the world, in Africa and in Asia. One of the features of the development of dementia is a loss of brain tissue. And what we see in individuals as they get older and increase their risk of dementia. Sorry, I just lost one slide. Let me go back one. Thank you. As people get older, brain volume decreases. And what you're looking at here are MRI scans of over 500 individuals across the whole of the adult age range. And you're looking at the amount of grey matter in the brain. And what you see is this progressive loss of brain volume with age. What's clear is that those people who are at greater risk of developing dementia have greater rates of loss of brain volume. And in a study carried out in Cambridge, where they examined people of healthy weight, an example shown on the left, and people who are obese, an example shown on the right, brain volume was lower in those people who were obese. So for people in their 50s, the people who were obese had a brain age which was 10 years greater than their lean counterparts. So this may be one of the mechanisms through which obesity is driving the increased risk of dementia. Dementia research has been growing rapidly over the last 10 years, along with the realization of the public health problem of dementia. And it's clear that so far we don't have good pharmaceutical agents to treat or to prevent dementia. And so improved lifestyle is central to dementia prevention. And that includes improved diet, reduced obesity, and more physical activity. Let me now turn to nutrition and the gut microbiome. And most of you, I'm sure, will be aware of the rapid advances in work on the microbiome. It's touching almost every area of biology and medicine. And this has been driven by improvements in technology, improvements in sequencing methods, improvements in databases, well-curated databases of all the major bacterial species in the gut and in other areas of the body, and better bioinformatic tools. This has allowed research on the impact of diet on the gut microbiome, so we now have a better understanding of how diet influences the gut microbiome and on how that has consequential effects on a whole range of health outcomes. From the lung to the brain, to the musculature, to the cardiovascular system, all of these systems are influenced by our gut microbiome and then potentially impacted by diet. The JPI, Healthy Diet for Healthy Life, has been active in supporting work in this area. And I'm giving you here an example of one piece of work funded by the JPI. And this group of researchers investigated the effects of birth mode and diet on the microbiome and on the risk of childhood atopic disease. What became apparent was that how we entered the world, whether we were born naturally by vaginal delivery or whether we were born by cesarean delivery, influences our gut microbiome, as does the way in which we're fed as an infant. So whether we're fed by breast milk or by bottle feeding. And all of these things then impact on our likelihood of developing asthma and other childhood atopic diseases. The JPI has also funded other work in this area and are now moving into research carried out in sub-Saharan Africa in an area of Tanzania just south of Kilimanjaro. And this was an investigation of links between diet, metabolism, and immune function in people living in rural areas compared with people living in urban areas. And what the researchers did was to use a whole range of state-of-the-art technologies, including whole blood transcriptomics, ex vivo immune function measurements, and on-targeted plasma metabolomics to unravel the connections between diet, metabolism, and immune function. Here I'm just focusing in on effects on metabolism. And what you see here is a list of many of the major metabolites identified in blood, and then a mapping of the pathways in which those metabolites take place. And what you see with the red arrow, which is the urban areas, and the green arrow, the urban areas, is that the patterns of metabolites, the pathways in which those metabolites take part, were very different. So in the urban areas, there were more pathways related to bile acid metabolism and arachidonic acid metabolism, whereas in the rural areas, we had pathways related to amino acid metabolism. And all of this leads to a better understanding of the way in which the change in diet associated with urbanization and westernization drives changes in the pattern of metabolites in the body and our immune function, with consequential effects on our health. And in this particular study, the researchers were able to identify the links between those changes associated with urbanization and a greater risk of gut inflammation. And gut inflammation, in turn, is related to a whole range of other adverse consequences. Let me then turn to personalized nutrition. As I mentioned in the introduction, 10 years ago, there had been a number of review articles drawing attention to the opportunities to use personalized approaches to improve the delivery of nutritional advice, but there have been very few intervention studies. And the Food for Me study, in which I was involved, was one of the first large-scale multi-country intervention studies. So what is clear was that the approaches which had been used to changing diet within populations, which were largely one-size-fits-all approaches, were relatively ineffective. They produced relatively small changes over rather large timeframes. So it was very ineffective as a way of improving public health. And so the Food for Me study asked the question, if we put the individual in the center and develop interventions which are relevant to that intervention, to that individual, could we do better? Could we enable that individual to improve their diet? At the start of this decade, there was a great interest in the links between genetics, nutrition, and health. And so within the Food for Me study, we wanted to use genetic information. And we hypothesized that genetic information might help to identify the right diet for you and for me, and so help to personalize advice. And having that information might motivate us to adopt healthier eating patterns. So I'm showing you here one of the major outcomes from the Food for Me study. What you see in the red box is a comparison of the control group, the individuals who are given one-size-fits-all advice, standard national advice on improving their diet. And on the right-hand side, in the paler blue bar, you have the people in the personalized nutrition group. And what you see is that there was about a 50% increase in the improvements in eating pattern in those people randomized to personalized nutrition. Now, outside the red box, to the right of the red box, you have the three types of personalized nutrition that we tested. We tested personalization based on diet alone, diet and phenotype, and diet, phenotype, and genotype. And what we discovered was that the type of personalization didn't matter. And there were no significant differences between the type of personalization, but overall, personalization improved eating patterns. So the take-home message from Food for Me was that personalized nutrition works. In this study, we saw no advantage of using more sophisticated data, for example, phenotypic or genetic information. And we had used the internet to develop and deliver this intervention across 7 countries in Europe. And so this provided proof of principle that we might be able to scale up this kind of intervention to improve public health at large. Work on personalized nutrition continues, and in the last several years, there's been a great deal of interest in using personalized nutrition to manage blood glucose concentrations. You may be aware that having large excursions, large changes in blood glucose concentration after a meal is associated with poorer health, with a greater likelihood of insulin resistance and potentially diabetes. So a lot of interest in how we might be able to personalize interventions to manage glycemia. And here I'm showing you some data from the recently published PREDICT study, in which they looked at some of the factors which influence changes in blood glucose concentration after a meal. And they looked at both breakfast and lunch, and looked at what the factors were which influenced how an individual responded to those meals. On the left-hand side of this slide, you see the individual lines show the individual changes in blood glucose concentration over time for all the people in the PREDICT study. And there's an enormous range of changes in glycemia. Some people showing very big changes and some people relatively small changes. On the right-hand side, then you see in the bar chart the contributors to those inter-individual changes in glycemic responses. And what it shows you is that many different factors contribute to those inter-individual glycemic responses. Of course, meal composition matters, so does genetics, so does medication use, age, and so on. So genetics is one factor, but it's only one of a number. So if we want to look at personalizing interventions to help people improve their glycemia, we need to look beyond genetics and look at this wider range of individual characteristics. I'd now like to turn to work that's been done on the developing of better tools and technologies, because all sciences, including nutrition, depend on having excellent tools to address whatever questions we want to address. 10 years ago, nutrition researchers had a problem that not many of us like to talk about, and this was the problem of measuring dietary intake. Very fundamental to nutrition research, but a really tricky problem. It was burdensome for researchers, it was burdensome for the participants in studies, and there was great uncertainty about the reliability of the data. In the last 10 years, we've made really important improvements in the measurement of dietary intake. Some of those improvements have come through using digital technologies, for example, using web-based technologies for 24-hour dietary recalls. And here I'm showing you an example of Intake 24, which has been developed by some of my colleagues in Newcastle. There's also been rapid development in the use of wearable sensing technologies, which identify not only what was eaten, but when it was eaten and some of the other circumstances around the eating event. By having the information in digital forms, and relatively easy to integrate that information with other data sets, for example, genomics information or phenotypic information or health outcomes, and begin to use nutritional information in much more exciting ways. There's also been another development which I think is even more important, and that is an attempt to escape from the tyranny of measuring dietary intake at all. And this required a completely new paradigm. And this depends on the concept that all food contain metabolites. Many of these are quite distinctive for the individual foods. So we can collect samples of body fluids such as blood or urine and look for food-derived metabolites. Now, these metabolites are not necessarily exactly the same as those that were eaten or drunk, because those food-derived metabolites are metabolized in three particular areas: in the small intestinal mucosa, by the bacteria in the large bowel, and of course in the liver. But we can use that information to translate the metabolites that we identify in blood or urine to tell us about what's been eaten. And the JPI has funded an important study in this area. That's the Food Biomarker Alliance, or FoodBall, which is led by Professor Ada Theskins, which set out to use metabolomics to discover biomarkers of food intake. This has been a highly successful project, and it parallels a number of other investigations going on globally, so that we now have a number of biomarkers for a wide range of foods. I'm just having a little bit of trouble with my, here we are. I hope you can now see the slide which says candidate biomarkers for multiple foods. We now have a wide range of candidate biomarkers for a whole range of foods, and I've shown you a few examples here. This collection of biomarkers is growing all the time. One of the continuing issues is about specificity, and I suspect that in the long run, what we will need is to use panels of biomarkers to get greater specificity. The JPI initiative, the FoodBall initiative, has also been important in developing a whole series of databases, including FoodDB and Exposome Explorer, and all of these databases will provide a strong underpinning for continuing research in nutrition. Of course, individual foods matter and they link to health outcomes, but the overall diet is probably more important. So we wondered whether we could use metabolomics to distinguish between overall diets which were more healthy or less healthy. What you see in the next slide sorry, having some problems. Let me go back one. What you see here is the outcome from a study in which a number of participants were fed a range of diets based on WHO, World Health Organization recommendations, from a very healthy diet down to an unhealthy diet. And the plot shows you the pattern of metabolites for these four diets. The least healthy diet is on the left and the most healthy diet on the right. And what you can see is that the pattern of metabolites can help us to distinguish between the overall healthiness of the whole diet. This is a very exciting area of development and something which I think will be something to take forward in the next 10 years. So another area of development which has been very important over the last decade has been the use of large, well-characterized prospective cohort studies. These have allowed nutritionists access to very large amounts of data on individuals, where we have not only data on dietary intake, data on physical activity, but also a wide range of other genetic and phenotypic data. And because these studies are carried out over long periods in large numbers, we can link them to a whole range of health outcomes. I'm giving you one example here. I'm sorry, this is very slow to move the slides on. Let's see if we can do that. So what I'm showing here is some data using the UK Biobank, which is one of these large prospective cohort studies, showing you that normal activities, this is cycling to work, is a strong predictor of the likelihood of developing cardiovascular disease and cancer. And this kind of large-scale prospective cohort work with well-phenotyped individuals can underpin public health nutrition interventions. We can also use that kind of information to look at links between lifestyle and genetics. And this again is using UK Biobank data to show that a better lifestyle can overcome the effects of genetics on dementia risk. And in this particular study, individuals were grouped according to genetic risk, and then their lifestyle, so whether they were a smoker, the physical activity, the healthiness of their diet, and alcohol consumption was measured. And what you can see is that the healthier their lifestyle, the lower was the risk of dementia at all levels of genetic risk. So let's now turn to the final part of my presentation, which is a glimpse into the future. I think in the future we'll continue to see rapid improvements in work on the microbiome, not only the gut microbiome, but the other microbiomes in and on the human body. Work is already underway to try and define a healthy gut microbiome, but that's proving really difficult because of the individualized nature of the gut microbiome. I think the concept, the healthy forest analogy, is very helpful in trying to understand the complexity of the gut microbiome. We can have a healthy forest which has a very different makeup. So just knowing which trees are present doesn't tell us whether the forest is healthy. We need to understand what it's doing. And the same is true for the gut microbiome. Up to now, most of the work linking nutrition with the gut microbiome and with health outcomes has been associative. And I think in the next decade, we'll see a real opportunity to move to studies of causality. We already have some examples of that, and I'm just going to show you one of those now. And this is a paper published very recently in the New England Journal of Medicine, in which the researchers used food intervention in undernourished Bangladeshi children. Now, this wasn't just any food intervention. This was a food intervention which was targeted specifically at the gut microbes. And the hypothesis was that by improving the healthiness of the gut microbiome, as well as providing nutrients directly, you could improve the health and growth of these Bangladeshi children. What this shows is a bigger improvement in growth of the children who were randomized to the microbiome targeted food intervention compared with a conventional food intervention. The final area that I want to draw attention to is work on nutrition and brain health. And I think this is where we're likely to see major advances in the next decade. Poor mental health is something which is a life course issue. It's something which is there in parents and in the effects on their children. It influences performance at school. It influences during our working life, and it has major impact on us as we age. And among those mental health issues is depression. There are more than 300 million people globally suffering from depression. It's a leading cause of disability, and it's a major contributor to the global burden of disease. Up to now, very little has been done on the links between nutrition and depression. We know, of course, that there's a reciprocal relationship between obesity and depression, and so there's likely to be important things where nutrition can help. We're likely to see big improvements in research on the gut-brain axis, in particular in the role of the microbiome in brain development and health of the brain at all stages of the life course. And there's already evidence for potential roles of the gut microbiome in a whole range of brain problems, including autism spectrum disorder, schizophrenia, problems of mood, and age-related brain degeneration. Of course, molecular and experimental nutrition research will continue to be important, and so we'll need to continue improving the models that we use for testing our nutritional hypotheses. And one example of an emerging technology is the growth of mini brains, little organoids that can be grown from human pluripotent stem cells. These mini brains recreate the 3D architecture of the human brain, and recapitulate the process of human neurodevelopment. And so they become a potential tool for testing nutritional hypotheses. So in this very personal view of the last 10 years and into the future, what I've tried to do is to tell you a little bit about links between nutrition and some important health outcomes, including diabetes and dementia, talk about the exciting work on nutrition and the microbiome, and indicate that's likely to be a continuing area well into the future. Talked about the developments of better tools and technologies, and those will continue to be important to underpin what we can do in research. And looking to the future, I think the real opportunity is to get deeper into our understanding of nutrition and the brain at all stages of the life course. Thank you very much indeed.
Thank you so much. And listening to your talk, but also looking at questions raised in the chat, it was such a dense conversation with such a lot of information for the audience. So we have recorded this event, so people can look back to it, which I think is helpful to really digest all the information that you shared with us. But I would really like, if you look at the chat now, I don't know whether you can see it. People really appreciate your talk, so that is brilliant. But maybe to highlight some of the issues raised is one of the questions is what is more important in determining the immune function and immune system development? Is it diet or is it the contact with germs, as both relate to urban and rural development?
Yeah, I mean, that is a great question. And of course, for these kind of questions, there's never a simple yes-no answer. And it's very much context dependent. And it's clear that the wider environmental exposure, which some people call the exposome, which includes that exposure to all kinds of environmental contaminants, to microbes, to our pets, as well as diet and so on, all of those impact on the development of the immune system. Of course, I focus very much here on the nutritional part of it, but I'm well aware, and I think the researchers are also very well aware, that that's only one part of a bigger issue. And if there had been time, I would have said a little bit about how nutrition fits into that bigger picture. So maybe this is an opportunity to do that. Because I think we as nutrition researchers need to embrace those wider areas of research and to recognize that we will not solve all these health problems alone. We need to work as multidisciplinary teams to solve real-world problems. And I think the real future for nutrition is to be a core part of all of those teams, which are bringing together researchers who are interested in a common problem, but from different perspectives. And I think that is something which will also drive the way in which we do our research in the next decade.
Yeah, I really recognize that because I just recently switched jobs, and I'm currently the new director of the Institute on Injury Prevention in the Netherlands. And for instance, with prevention of physical injuries, diet and nutrition status is actually very important. So even moving out of nutrition, it remains a very important part of the work. Yeah. So these multifactorial approaches, I think, are essential.
And it connects very much with some of the things we heard yesterday from in the first plenary presentation, the first talk of the day, looking at the sustainability issues, because again, it links into that work on planetary health. So we can't separate what we do on human health from health of the planet. And I think that's going to be a challenge for nutrition researchers as well.
Mm-hmm. Maybe another question. Somebody asked, also with you putting a lot of emphasis on the microbiome and trying to influence the microbiome, how stable is gut microbiome if it's modified, and does it revert to its original state?
So another great question, and there's a great deal of individuality in the gut microbiome, but it's been clear that for most people, their gut microbiome is relatively stable through adulthood. Of course, it can be changed by things like going on a course of antibiotics or changing diet and so on, going on holiday, those kind of things. But generally, our gut microbiome returns to similar patterns to what it was before. But there are subtle and important changes over time, and it's capturing those changes over time which has been a major challenge. Most of the studies that we have been cross-sectional, and what we really need are longitudinal studies where we measure the same individual, not just over a few weeks or a few months, but over years. And we only just now begin to get that longitudinal data, so we can begin to answer the question about what are the important factors which change individual gut microbiomes and the extent to which we can manipulate that in positive, healthy ways.
Mm-hmm. Maybe a quite different kind of question, because there are a lot of really content detailed questions on the research and the science. But we have a question from Hans-Jörg Lutzeyer, who says, do you see any progress in integrating nutrition into health professional higher education? Since we acknowledge that nutrition plays such an important part, do you actually see that it makes its way to the education of health professionals?
Well, I think the short answer is not as fast or as far as we would like. It's happening. And in the UK, for example, all medical students have to have a certain amount of nutrition, but it's not nearly enough. But the very positive thing, and I think this is something which we should be very encouraged by, and that is that the students themselves are asking for more nutritional training. They recognize that it's something that they need to know about, that it really matters to understanding health. And so students themselves are asking for more nutrition. And I think that will persuade the rather conservative medical education authorities to find creative ways of changing the curriculum to include more nutrition. And it's happening already, for example, by bringing nutrition into case studies. So instead of it just being case studies where you get a whole load of lab results on things they've measured, some biochemical metabolites or something, you now get information on eating patterns and so on to try and understand more about the person and their circumstances. Mm-hmm. So I think there's great opportunity to do it. It needs to happen, but it's not happening nearly fast enough.
Okay. And I think what I also really liked about your talk, specifically if you look at the future, the focus which should be put on research related to the nutrition and the brain and mental health and stuff like that. I think that is also a really interesting new area of research. Yeah.
I think it's a very under-researched area. And if you do a literature search on it, you'll find that there are very few experimental studies. There are lots of descriptive studies, and what we need are well-designed studies which really start to test some hypotheses in this area. But I think we've now got an opportunity to do that. We've got better tools for doing it, and we have an entree into that area which wouldn't have been possible a decade ago.
Okay. Well, I would like to. It is already 3:00. Time flies, especially if there's such an interesting conversation going on. And I think also when you look at the questions which were raised, there is still a lot of, if we would have been in an environment where we could actually chat with each other, people would have run to you during the coffee breaks and asked you all these detailed questions. So maybe we can come up with some kind of interview or whatever, to put on a website to highlight some of the issues raised. So if you're open to that, that would be brilliant.
Very happy to do that. And of course, people can email me afterwards and I'm happy to answer questions.
Okay, thank you so much. And if you would have been on stage, I would have, provided you with a nice gift to thank you for this presentation. It will be on the mail, so don't worry, we won't forget you, and we will thank you very much. So thanks for this very nice start of the second day of the conference. We will have a small break now, and then we will start at 3:15. With the workshops. People have already indicated which workshops they want to attend. So at 3:15, people are supposed to, enter the room for that workshop. Put on their cameras so that people can really have interactive sessions. And then we will be back in the lounge at 4:30 to finish up the day. So thanks again. Thanks so much. And
Thank you very much.
We will remain engaged. Thank you. So for everybody, you can switch off. You can go back to the lobby and then enter the session you would like to attend the workshop at 3:15. So enjoy your afternoon.